Okay, it's noon, so let's get started. Lots of stuff to cover this week. So instead of doing a whole recap of what we talked about on Friday, because I know you guys still all remember that, that's all we thought about this whole weekend. Let's see, the last part, I really kind of want to focus on that terminology again that we finished with. So at the end we talked about spermatogenesis, right, which really makes making sperm. So can you name a particular germ cell type in a testis? That's an easy one. Okay, can you name me a kind of a undergraduate college student?
Freshman.
Freshman, there we go. Thanks, Ethan. So now let's do one for germ cells.
Spermatogonia.
Yeah, spermatogonia. Let's do that one. Spermatogonia. I like spermatogonia. Perfect. Spermatogonia. What came after spermatogonia? Those were the baby ones. Something that finishes with sites? Spermatocytes? Perfect. Yes, close enough. Close enough. Primary and secondary spermatocytes. So what's the difference between a primary spermatocyte and a secondary spermatocyte, except for the word primary and secondary?
Meiosis.
Meiosis. So which one does meiosis?
Meiosis 1 is primary.
Meiosis 1 is primary, and
Secondary is meiosis 2.
yeah, and secondary spermatocyte is meiosis 2. And then we are spermatid, and then finally sperm, right? Okay, so how many of you enjoyed and thought, you know, in high school and maybe some other freshman biology class, that meiosis is the coolest thing ever? And we really need to talk about this more and more. It was okay. It wasn't the worst thing. Yeah, I know. I hated physics. I've never passed a physics class in my life, not even in college. So if you ever have an ambition on becoming a faculty professor, unless it's in physics, you don't need it, okay? Anyway, okay, meiosis. So I want to make, because in order, later on when we start talking about what happens in the female, we really need to kind of have an understanding of what happens with meiosis 1 and what happens with meiosis 2, okay? So, that is a cell. Let's see, what's your favorite number between 1 and 22?
7.
Chromosome number 7, that's what it's going to be. Okay. How many chromosome number 7s do we have? 2, right? One from mom, one from dad. Okay, here we go. We're going to focus only on chromosome 7. We'll just ignore the rest. Okay. There's chromosome 7. And there's chromosome 7. One from mom and one from dad. Now, when we do mitosis, or cell proliferation, or meiosis, what is the first thing that has to happen with chromosomes? Or actually not chromosomes, with DNA. Unwind, yeah, it's something, let's do it this way. Yeah, supercondensing, but it's something, let's see, if the whole point of meiosis is doing what? Why do we need to do meiosis?
DNA.
Okay, so let's go back, ignore my question. You have the answer to my previous one. DNA replication, duplication, okay? When we divide, before we do that, we need to duplicate our DNA amount. Otherwise, every division will lose stuff, right? So, this is what a chromosome looks like. Now we do DNA replication. How many chromosomes do I have here? Yeah, we always get that, 4. No, we don't. We're not making more chromosomes. Okay? How many chromosomes of dad do I have there? 1. We have 1 chromosome 7 from dad and 1 chromosome 7 from mom. Now, this is a chromosome. Okay? This is a chromosome, but a chromosome after DNA duplication or replication has 2 chromatids. Okay? So this is still 1 chromosome, but it has 2 chromatids. We just need to give it, these things a different name. That's it. But it's still 1 chromosome, okay? So here's 7, 7, this is mom, this is dad, okay? Now normally what happens when we do mitosis, all these chromosomes line up like this, all underneath each other, right? We do that for all of them, all 46, okay? And then we split these, so they're all like this, all lined up. This goes that way, that goes that way, right? We're separating the chromatids, and then we're good again, right? We still have 46. I haven't changed numbers here. It's still 46. Make sense? Okay, so meiosis 1 is the odd thing. Because it's kind of lining up like this, it's going to line up like this for all the pairs. Chromosome 7 from mom and chromosome 7 from dad are going to line up and hold hands. Okay? Chromosome 8 from mom and dad, they're going to line up and hold hands. So they're lining up like this. They're not so shy anymore. They know what's going to happen. Okay. And now they're holding hands, and we get crossover, swap genetic material between homologous chromosomes. Homologous is just a fancy word for the same. 7 and 7, 8 and 8. Okay. So once you do this for all of these, and again for all these 23 pairs, okay, so this is what happens in a primary spermatocyte, right? And now we're going to split. What happens after meiosis 1? Meiosis 2. How do we call a male germ cell that does meiosis 2? Secondary spermatocyte. So 1 primary spermatocyte makes how many secondary? 2. Okay. So what does each one have? Well, I'm just highlighting one of them. There you go, chromosome 7. I'm only showing chromosome 7. Yeah, so the swapping, the holding hands, is a crossover. The crossing over genetic material. So now I cannot call this dad and mom's chromosome anymore, right? Because we swapped genetic material. It's mom and dad is here, and mom and dad is there. Okay? So and we have this for all 23. Chromosome 1 through 22, and then X and Y. Okay? So there's 23 chromosomes in the secondary spermatocyte. But each chromosome has how many chromatids? 2. Okay, so still twice the amount of DNA. So then this one splits, and this one too. And meiosis 2 is really kind of like mitosis. What happens with mitosis? We're separating these two, right? This is the same thing here. We're separating right across the middle there. So this one has 1. Here's another one for chromosome 7. Here's 1. Here's another one. Make sense? So for meiosis in reproductive biology, when we talk about this, I just want you to kind of picture what's happening with these chromosomes. That's it. So from 1 primary spermatocyte, how many spermatids do I get? It's on the board. One primary spermatocyte, 2 secondary spermatocytes. Count with me. 1, 2, thank you, 4, 3, and 4. Make sense? I know. It's Monday. And plus it's the end of the semester. It's like, oh God, this is still, the end is in sight. There's light at the end of the tunnel. We're almost there. Almost there. Okay, does this kind of make sense a little bit? Perfect, so that's it for meiosis, okay? Now, when we talk about female, right, This just continuously happens in males. You just go, 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 there's no stopping. Remember last week when we talked about what happens in females during fetal life in the ovary? They arrest in meiosis 1. Specifically, they arrest when they're like this. Okay? And they're stuck like that. When you're born, baby girl is born, that's how her eggs are. When she's 8 years old, that's how her eggs are. When she's at puberty and we start, then some of them will actually do this. Not all of them. Okay, we'll talk about that on Wednesday. Okay? Any questions about this stuff? Any questions about male reproduction or this stuff? Does it kind of make sense? So if this is a spermatid, what's the last part? How did we call that? Metamorphosis, a round cell into an alien, a spermatozoa or sperm or something like that. Okay? Okay, let's finish male reproduction. So, just a slide. There's not that much on this slide that wasn't on the last slide that we finished with last week. Here are the different germ cell stages. Kind of a quick recap again. We start with the basement membrane. There's a lumen on top. And the Sertoli cell was this crazy looking cell with all these kind of like pockets left and right. And the Sertoli cell kind of extends from here. This is the basement side, the basal side. And it goes all the way to the top. That is one Sertoli cell. And there's another one right here, another Sertoli cell. Okay? And then what happens here, what I highlighted, is that each Sertoli cell holds hands with each other. They're all holding hands. Okay? And those are tight junctions, very specialized, unique tight junctions. Okay? And on this slide, what you see in the top right here is basically a cross-section of a seminiferous tubule where we got rid of everything except for 5 Sertoli cells. Okay? And these 5 Sertoli cells are holding hands right there. Okay? But you can picture it going all the way around. In between the Sertoli cells, we have the germ cells. So germ cells are never inside other cells. They're never inside Sertoli cells. There's no such thing as a cell inside another cell. Okay? The germ cells are all in between the Sertoli cells. And on all sides, this is just one-dimensional. Okay? But figure it goes behind and on top and all over the place. Okay? And by holding hands like this, they're actually forming what is called the blood-testis barrier. Okay? So they form a barrier. They also form, if you kind of just look at this figure, basically 2 compartments. There's an area between two Sertoli cells right here, that is called the basal compartment. That is basically the basement membrane up until where they're holding hands. And then there's also an area for germ cell that goes from the blood-testis barrier all the way to the lumen. More space to hold some germ cells. Okay. So the blood-testis barrier is something that is extremely dynamic because think of it as a blood-brain barrier. The blood-brain barrier is very important, right? What's the function of a blood-brain barrier? Very simple. Do we want everything that's in our bloodstream, do we want it to go to our brain? Yes or no? No, so keep things out of the brain. Okay? This is kind of the same thing. The stuff in blood, don't want it. Okay? Now, the only difference between the blood-brain barrier and the blood-testis barrier is that the blood-testis barrier is very dynamic. It needs to be able to open and close, like a sliding door. Okay? Because those germ cells that are in this compartment right here, at some point, they need to be able to go through this door over here into the lumen. Right? Now, let's think a little bit. We talked about all these different germ cells that we have. Right? So which one can I find in the basal, the basal room, like this basal compartment right here? Spermatogonia, right? When we did this analogy with freshmen and sophomore and stuff, where did we chuck the freshmen? In the basement. Okay? So the spermatogonia that divide, okay, do mitosis, they're all in that basal compartment. What kind of germ cells can I find in that second room after the blood-testis barrier? Yeah, primary spermatocyte, secondary spermatocyte. Basically, what are these germ cells doing? Meiosis. So the germ cells that start doing meiosis and all that stuff and become haploids, they're all in this compartment right there. Okay? So why is this important? Why do we need to set it up this way? What do you think would happen if the blood-testis barrier was not working, like it's kind of open? I'll give you a hint. When does sperm first made in men? When's the first time men start making sperm? Puberty. Okay. Now let's think about it a little bit. What would happen if the blood-testis barrier was not functioning quite well, it was letting stuff in?
Is it to regulate hormone control into the testes?
Yeah, the hormone control, but where in this, so in this scenario, where are most of the hormones being produced? Is it in here or is it more outside? Leydig cells are outside, right? So when is the first time inside a boy's body we have a primary spermatocyte? Puberty? Okay, so when does puberty, what are we talking about? When is that?
11, 12.
Yeah, 11, 12, 13, whatever. Okay? So when you're 12 years old, let's say 12, that's the first time you make a cell that is doing meiosis. How do you think your body would react to that? How does the body react to foreign things? Kills them, immune response, okay? Inflammation, all that stuff. Your body, or at least in a man or a boy, has never seen that cell before. When does your immune system develop? Well, not at puberty. Okay, you have not lived for 13 years without an immune system. Okay, so the one thing that does happen is if that blood-testis barrier doesn't work well, yeah, an inflammatory response, a huge one. Okay, your body will start attacking primary spermatocytes, secondary spermatocytes, spermatids, sperm, all foreign stuff. Okay. Now the other thing that I kind of want you to think about is that basically with these two compartments, you're providing two homes. You have one home, a basal room, for cells that do mitosis. You have a second room where you provide nutrients for cells that do meiosis. These are all different things that these cells need. By making these two rooms, you can actually secrete stuff in that room that the cells need. Does that kind of make sense? So it doesn't diffuse out all over the system. Okay? So really important, a basal compartment and an adluminal compartment. Okay? Make sense, or is this like completely like, what the heck is he talking about? It's Monday.
So then, if you got an infection in your testes, would your immune response not be able to do, or would your immune system not be able to do anything?
Yeah, so it depends on where the infection is. Like, so the only thing that is immune privileged is this part right here, right? Your immune, so the blood is outside here. Leydig cells see stuff from blood. Sertoli cells can get stuff from blood, and they have to because we have hormones coming from the brain that act on these cells. Immune cells will act on this stuff too, but the part that the immune cells do not get access to is that little area right there. Okay? Cool. Make sense? Yes? Two. Go for it. So it's not necessarily a specific name for it, it's just an inflammatory response, but there's certain, for example, toxins, especially metals. So any kind of exposure to high levels of cadmium, for example, if it becomes toxic, it actually destroys some of the cytoskeletal elements in the Sertoli cells, but it can also actually disrupt the blood-testis barrier. They become more open. Response would be an inflammation. Okay. On top of that, too, is you start losing all your germ cells, right? If the Sertoli cells do not maintain their shape and their structure, all that stuff starts sloughing off into the lumen, and it's gone. Okay. Okay, let's move on. So this is kind of, again, going back to a picture of what a cross-section of all these Sertoli cells look like. And it may not be that horrendously obvious when you look at the top left. Like maybe it looks all the same. But if you look a little closer, maybe you recognize that the cross section here, we see actually some sperm tails and stuff in here, but here not so much. And basically what you just start thinking about is the fact that if it will take 64 days to go from being a freshman to a senior, okay? 64 days to go from a spermatogonia to being a sperm. If it takes 64 days to make sperm, do men have a 64-day cycle? Can they release sperm every 2 months? Maybe that's a good thing or not, I don't know, but does it or not? No, then what the heck is going on? Any thoughts, ideas?
Probably a storage space.
Sorry?
There's probably a storage space.
It's probably a storage place. Okay, that's good. It could be. There could be a storage place. Let's go with that just for a second, okay? Storage place. So let's build on that a little bit. Boys make sperm the first time when they hit puberty. Let's say 12 or 13 years old. When do men stop making sperm? When they die. Okay, so let's say they're healthy individuals and let's say 80 years old. Okay? Wow, that's many, many years of making sperm, right? So some of these cells that are in the basal room, the spermatogonia, are what are called stem cells. What are stem cells? How many of you have heard of stem cells? Go for it.
There's cells that haven't been differentiated yet.
Okay, so that's good. That's kind of one part of it. Those are cells that haven't differentiated yet. Another part of that's very important for a cell to be a stem cell is something else. If, let's say, from 13 to 80, what is that, 67 years old? For 67 years, how many sperm are being made? So let's say in an ejaculate we had how much sperm? 50 to 400 million sperm. Well, our testicles, when they're very young, the size of gigantic bowling balls to hold all that sperm. Yeah, but if we talk about millions of sperm, and it's not every 60 days either, it's like the whole time. So what's happening? That's the second part of the definition of a stem cell. What is that? So here you are, a stem cell. You can divide, you cause two daughter cells. One becomes a cell that becomes more differentiated, so I'm skipping a whole bunch of stages here. What's the other part? Self-renew. They have to be able to give a copy of themselves. Otherwise, it's not a stem cell. Okay? So they have to be able to replace themselves. So you always have a pool present. That's why men, when they're 80 years old, they can still make sperm. And that's also why you don't have to have testicles this big to hold billions of sperm to provide a lifetime supply of sperm. Okay, some of these spermatogonia, not all, some of them are actually stem cells. So when they divide, they give a daughter cell that will differentiate, and they will self-renew. Okay? Do you have something like that in the ovary? No? How do you know? Everybody should know this. How do you know that there are no stem cells like that in the ovary?
You arrest at meiosis 1.
They arrest in meiosis 1. Excellent. So they're all stuck in meiosis 1. What's the other thing? When women get older. Menopause? What is menopause? Women getting really crabby and crazy? No,
You can't say that.
not like that. No, no. Okay? They don't ovulate anymore. Okay, so at that point, there's so few eggs, you're not kick-starting another cycle, and it's because you kind of run out of your supply of eggs. Okay? Now, if there were stem cells, then why would you be running out of eggs? Make sense? Okay, going back to, well, if it takes 64 days to make sperm, how come we don't have a 64-day cycle? Kind of look at these adjacent tubules, right? Do we have all the same stages present at the same time? That's the thing. So basically what I want you to think about is if you have a tube like this, longitudinal, that at some area right here, this is maybe where you're making sperm. Okay? In this area right here, you may have the freshmen and the sophomores, and that's it. You haven't started meiosis yet. Okay, so this is kind of really extreme. Maybe another way of looking at it is this slide, where this is for rat, this is mice. It doesn't matter. All, humans have it too. But basically, one cross-section, you'll have all these generations of germ cells. Another cross-section, you'll have this. Another cross-section, you have something else. It is asynchronous. Asynchronous, meaning we don't have sperm production throughout every single part of the seminiferous tubule throughout your whole testis. Does that make sense? So it's just kind of all at different places. There's some sperm there, there, there. 2 days later, there's some sperm there now and not there anymore. Does that kind of make sense? So it's more continuous but asynchronous. And this slide, ignore this slide. Okay. Any questions about this stuff? Kind of make sense? Cool. Okay, so the last part is metamorphosis, spermiogenesis, and this is really where we have a spermatid. How many chromosomes in a spermatid? 23, perfect, 23. How do they look like? This or this? The one on the left, right? We separated out the sister chromatids, the chromatids are gone. Now we have a single straight chromosome, 23, okay? And basically a sperm now, that round cell that turns into something with a head, a midpiece, and a long tail. The head has two things that are of interest to us. One is a nucleus, and at this point the 23 chromosomes become highly compacted. Okay? Make it really small. We replace certain things. I think maybe Dr. Warren in the beginning of class talked about something called histones. DNA is wrapped around histones. Ignore this part, but histones are gone. We come up with different things to highly compact all that DNA. Okay? And then we have something called acrosomes. Think of acrosomes as a large garbage bag filled with enzymes. Okay? Filled with enzymes. And we'll talk about acrosomes again when we talk about fertilization on Thursday. Okay? Because that part, that bag with enzymes plays a role in fertilization. Okay? And then we have, you can kind of ignore the midpiece, the centrioles. The centrioles is more for, they're anchors for microtubules. They're important for dividing, cells dividing and stuff like that. Just ignore that. But then we have mitochondria. So we have a midpiece with lots of mitochondria that are kind of coiled around, starting a microtubule, which is the tail. What is the importance of all these mitochondria in that midpiece?
They allow for movement, the energy.
Yeah, kind of the energy for the tail. Okay. Now, the one disclaimer I have to make here that sperm do not move their tails or are not swimming when they move through the male reproductive tract. Think about this. These are male germ cells that are just really lazy. The movement of sperm down the male reproductive tract is through smooth muscle contraction, lining the male reproductive tract. Sperm are not swimming. They will never swim in the male reproductive tract. Okay? So at some point they're deposited in the female reproductive tract, in the vagina, and then we have to move through the vagina, the cervix, the uterus, okay, all that stuff. They're still not swimming. Still not beating their tails. This time, it's the female reproductive tract with smooth muscle contractions that help move the sperm up. Okay? The sperm will only start beating its tail and using up all these mitochondria when it gets really close to the ovulated egg. It's almost like they can sense where the egg is at, they get super excited, and they start beating their tail like crazy. Okay? And we'll talk about that on Thursday. Yes?
I was going to ask how they know.
How do they know? Yeah, we'll talk about it on Thursday. Okay? Sounds good? So even though we have all these mitochondria, we're not using them to swim all the way from the testis all the way to the ovulated egg. If I were to ask you right now, the mitochondria that you have, where did they come from? Oh, your mom. So what the heck is going on?
This mitochondria does not contribute to the baby, just to get the genetic information into the cell.
Yeah, so you're right. And so there's actually two kind of thoughts here. One, we know that these mitochondria get a protein tag. It's called ubiquitin, just don't write it, that doesn't matter. And basically these male mitochondria that are in sperm get a tag, and then once they're in the egg, they get destroyed. Okay? Some people believe that this has to do with these mitochondria because there's that hyperactive motility when they start going all nutty because of the ovulated egg. They generate so many reactive oxygen species, they cause damage to the mitochondrial genome. And that may be dangerous or something like that. That is one school of thought. The other one is really that if you compare the size of that egg with its cytoplasm and lots of mitochondria, and that tiny, tiny little sperm, and yeah, for that tiny little sperm, there's quite a few mitochondria, but you put that into the egg, I mean, those mitochondria are just kind of diluted out there. You just never find them again. So those are kind of the two trains of thoughts that are out there. Okay, and then we have a tail with microtubules and swimming. Okay, let's do hormonal regulation of male reproduction, which is really easy. So, always remember the reproduction starts in the brain, hypothalamus, GnRH. Okay, then next it goes to the anterior pituitary gland. Then we have two gonadotropins. Gonado, because gonad, it acts on gonads. Trophic means these are trophic hormones, the nourishing hormones. Okay, so if you start with LH, comes from the anterior pituitary gland, hits the bloodstream, travels to the testes, and that one acts on Leydig cells. Okay, on Leydig cells. And Leydig cells, we all remember from last week, are the steroid-producing cells in the testes, so they secrete testosterone. Perfect. Okay? So they make testosterone. Okay? Now let's do the other one. Okay, we have another hormone coming from the pituitary gland, FSH, follicle-stimulating hormone, a stupid name in male reproduction because there are no follicles, but it doesn't matter, FSH. Okay? Acts on Sertoli cells. Okay, and when FSH acts on Sertoli cells, it does a couple of things. Okay, the first thing it does, it causes Sertoli cells to secrete a hormone called inhibin, and this is one of those things that I wrote on that slide called Sertoli cells. One of the functions was because Sertoli cells secrete inhibin. Inhibin causes negative feedback to the brain. Endocrine systems always have a negative feedback. Okay, so we can put a brake on things, so we can start again and again. Okay. The second thing that FSH does to Sertoli cells, it tells the Sertoli cells to secrete into the lumen, so where we put sperm as well, something called androgen binding protein. I also put that on that one slide called Sertoli cells. Androgen binding protein is a protein that gets dumped into the lumen where sperm is as well. And what do you think it does there? Based on the word. Binds androgens, binds testosterone. That's what it is. Okay? And then finally, you need FSH and testosterone together in order to do spermatogenesis. You can only make sperm in a testis if you have FSH and testosterone. If you have a mutation in FSH receptor, you don't make sperm. If you have a mutation in the androgen receptor, you cannot make sperm. You need both. Otherwise, it's not happening. Okay? So, this testosterone secreted by Leydig cells, where do you think it's going to go? Where are some places? One is very obvious because we call it the male sex hormone because of what? Why do we call it a male sex hormone?
The secondary sex development?
Yeah, that too. And if we were to take blood samples here from everybody in class, it's higher in males than it is in females. Good. Testosterone. Testosterone is higher than it is in females. It's higher in blood. It hits the bloodstream. Okay? During puberty, it enhances muscle development, causes facial hair growth, deepening of the voice, all these male secondary traits that you see starting at puberty. Okay? So it hits the bloodstream. When it hits the bloodstream, it causes negative feedback to the brain. Okay? So if you put testosterone into your bloodstream, goes to the brain and it shuts off LH and FSH. Okay? Testosterone also goes to the Sertoli cells and it goes to the lumen. So now we have testosterone in the lumen and this androgen binding protein in the lumen. Just think of this as androgen binding protein is a car and testosterone is the driver. Put them two together and basically now you have something that can drive along the male reproductive tract, exposing all that tissue to testosterone. From the lumen to all these epithelial cells. Kind of make sense? Awesome. Any questions about any of this stuff? Because that's really hormonal regulation of male reproduction. That's it. This goes on and on. It starts at puberty, and we've talked about this until you stop. Okay, all good? Shall we pick it up a little bit? Go a little faster? So, what happens if we apply exogenous testosterone? For example, anabolic steroids. Sometimes you see this with athletes, professional athletes. Why would professional athletes want to inject themselves with anabolic steroids?
We can grow their muscles larger.
Yeah, because of all these anabolic effects, and it promotes muscle growth and things like that. Yeah, it actually heals faster, but yeah. What does that do to reproduction? Now that we know the endocrine system. Sorry?
I think we'll stop production.
Yep, yeah, it stops spermatogenesis because of this, right? If you inject yourself with a potent synthetic androgen, it goes to the brain, it shuts off LH and FSH. If you shut off LH and FSH, you stop spermatogenesis. And if you do this for long enough periods of time, this is what the seminiferous tubules look like. They're devoid of germ cells, no more germ cells. Okay? Okay, so let's quickly move through the male reproductive tract. We have seminiferous tubules, efferent ducts. I kind of mentioned those. Just a kind of a footnote, efferent ducts are really important in male reproduction. That's where fluid absorption occurs. And you're basically concentrating sperm. If that does not happen, then basically the fluid all starts backing up to the testis and it causes atrophy or cell death. And the hormone that does this is estrogen. It's actually estrogen in this area of the male reproductive tract that is critical for male reproduction. Okay? So then we have the epididymis and the vas deferens. So the epididymis is just a long tube. We do divide it into a head, body, and tail. Key role for the epididymis is two things. One, store. You store sperm. Sperm gets stored there for a little while. The second function is sperm needs to mature there. Okay? If you take a sperm straight from the testis and you put it into a dish with eggs, it's not going to do anything. It is incapable of fertilizing an egg. So it has to reside and be stored in the epididymis for a while before it could ever fertilize an egg. Okay? Now, it says here acquire motility. Normally, inside the body, as I told you, the sperm is not swimming. It is just like if you were to take it out of the body, it would never be motile if you take it from the testis. It has to go through the epididymis. Okay? And then transport, result of muscular activity. That's kind of what I talked about, movement through the male reproductive tract. Vas deferens, just a long tube. Lots of muscle layers. Okay? That's really all it is, more storage. The seminal vesicles are the accessory sex glands. Okay? So here we have the seminal vesicles, prostate, bulbourethral gland. All these things provide just nutrients and buffers and things to help move sperm along, to keep the sperm happy, all that stuff. Okay? It says activate motility here, cross that out. Just ignore that part. Okay? That's it. You're experts in male reproductive physiology. Do we feel like we're experts or? Nah, we just want to survive. We're in survival mode. Everybody's in survival mode at this point. So, how many of you thought this was horrendously complicated, male reproduction, aside from terminology? Not too bad? Okay, not to scare you, but if you thought this was complicated, wait till we talk about female reproductive physiology. Okay. Okay, so this is kind of an outline. Okay, we're going to do the same thing. First a little bit of anatomy, and then we get into the hormones and all that stuff. Okay, so this is kind of the outline of female reproductive physiology. Female reproductive structures, okay. This is a way better picture of what the female uterus looks like, okay. No uterine horns. We have ovaries here, fallopian tube, or fallopian tube and oviduct is the same thing. And then we have this uterus, cervix, and the upper portion of the vagina. Now, the uterus, there's two parts to it. There's an inner lining, okay, which is right here, the inside. That is called the endometrium. We will talk about the endometrium on Wednesday. That is the part that sloughs off during menses, okay? And then outside you have this thick muscle layer. That's called the myometrium. The myometrium, that muscle layer, is involved with contractions during labor, okay? So the ovary is very similar to the adrenal gland in the sense in that you can divide it up into a cortex and a medulla. The medulla is the inside part. There's nothing interesting there, so we will not talk about this again. The outside, the cortex, has all these interesting structures. So we don't have tubules or things like that in the ovary. In the ovary, the interesting structures are follicles. So follicles are structures that contain the egg and some of those special somatic cells we talked about, like granulosa cells and theca cells. So they form all these different structures called follicles, okay? And you can find those on the outside in the cortex. This is the case for all mammals, all ovaries in mammals except for one. There's one mammal that has an ovary that's kind of like people refer to as an inside-out ovary, an inverted one. Anyone want to take a guess? Platypus. Same as last section, platypus. Did Dr. Ballern talk about platypuses a lot this semester? No? This is really an amazing thing. Lays eggs, has 5 X chromosomes, 5 Y chromosomes, gives fluorescent light. But the platypus is not it. It's really cool though. Everybody here has seen this animal that I'm talking about. And it's big and tall. Not a giraffe. I'm so happy that nobody's saying dinosaurs. A couple of semesters ago, people started talking about dinosaurs, like for Pete's sake, just a normal mammal thing. So how about we go with something that you can, cowboys ride on it. Not only cowboys, but a horse of course. There we go. So a female horse has an inside-out ovary. Okay, so I'm going to post a PDF with basically just words on it defining these different follicles. So for the final exam, you need to know these different types of follicles, okay? So what's going to happen is we have all these different structures. The first one is the primordial follicle. The primordial follicle is basically a baby egg arrested in meiosis 1, like this, okay? We call it a baby egg because it's not fully grown yet, okay? So this baby egg, we can also call it a primary oocyte. Just like the primary spermatocyte is in meiosis 1, a primary oocyte is in meiosis 1, okay? So this baby egg kind of grows into what is called a fully grown oocyte, okay? A primary follicle has a fully grown oocyte. Between a primary and a primordial follicle is that the egg is fully grown. Now, the egg is not getting bigger. The egg in this thing is the same size as the egg in this ginormous structure on the bottom, okay? We do see that there are more granulosa cells surrounding that primary oocyte that's fully grown. From a primary follicle, we go to a pre-antral follicle. The difference there is we make the granulosa cell bigger, more cells, okay? And then we start seeing a theca cell layer that starts to develop around these things as well. So that is called a pre-antral follicle. The pre-antral follicle continues to grow, and basically what we're doing, the egg is not getting bigger, still a primary oocyte, more granulosa cells, and the early antral follicle develops into a mature follicle, and what we see there is within that really thick granulosa cell layer, we're making a fluid-filled cavity called the antrum. So that antrum is taking up a lot of space. The antrum causes that follicle to become so big. Okay? So that the fluid-filled cavity also, if you kind of look at what happens to the granulosa cell layer, you see some of these granulosa, so here's the egg, you see some of the granulosa cells are still kind of close to the egg, and those are called cumulus cells. Okay? The cumulus cells are these eggs closely surrounding the egg. Okay, just to give the granulosa cells a slightly different name, cumulus cells. You also have granulosa cells that are all the way on the outside of this fluid-filled cavity, the antrum. Those are also granulosa cells, but they're further away from the egg. And those are called, so they're all the way here, okay? Those are called mural granulosa cells. Okay? So basically, because of this fluid-filled cavity, we're separating the granulosa cell layer into two parts. A couple of cells really close with the egg, cumulus cells, and granulosa cells that are all the way on the outside. And by the time you have this mature follicle, you have lots and lots of theca cells as well. Okay? So I'll make a PDF that will have all these different things defined as well. Like these cells go with this, these cells go with that, et cetera, et cetera. Okay? But what I want you to start thinking about is picture this in your head, because on Wednesday, when we start talking about the monthly cycle and what's happening, we're going to refer to what's happening with these and what's happening with this. Okay? So it helps if you kind of picture in your head what is there. Okay? That's it for today. Good luck the rest of Monday. Have fun on Tuesday, and I can't wait to see you on Wednesday.